So that's why, so maybe I am not that much of detail, but today we will be talking about, because today the responsibility has been given by Dr. Rajgopal sir, that membrane glycolipids in photosynthesis, thylakoid biogenesis, and chloroplast development. So very, very nice topic it is, and I was not much more aware of this one, but I could see that yes, it's a very, very interesting things which can be also give you some of the clues as well as some of these your ignitions in your thinking process to come out some of the good topics as well as questions. So if you look into this, the chloroplast, as you know that because last few days you are acquainted that it's a double membrane organelle and called the plastid, and which are unique to the plant and algal system. And perhaps Professor Takahashi was talking about the alga, means all the Chlamydomonas we are talking about as a, and they are capable of oxygenic photosynthesis, and of course it has a outer and inner envelope membrane, and chloroplast have extensively developed internal membrane system. That's what we talked in the last lecture, and where the photochemical and electron transport reactions generally takes place. So thousands of proteins and pigment as well as the photosynthetic factors, cofactors are available there, and they are embedded and in the bilayer formed by the glycolipids. So these bilayer membranes, which is a glycolipid in nature, and evolutionary, if you are looking at that chloroplast and cyanobacteria, they have the same lipid composition. So as a result, the non-phosphorus glycolipids are the major constituents of this membrane. And although the typical cell membrane in animals as well as the fungi or non-photosynthetic bacteria use the phospholipids for building blocks of the lipid bilayer. So as a result, what happens, this is a global phenomena, you can say, in a cell where the membranes are there. So, and as you know that there are stroma and grana, and both the outer and inner membrane, and we will be talking about this outer and inner membrane and what is the compositions of this one. If you are looking with the normal things that what the chlorophyll is doing for, because we are looking for these things here, that in the visible light, because all these phenomena or the chlorophyll activity is in the visible light spectrum, not in the far red or into the infrared region. So as a result, what happens that the visible part of the spectrum, it has the violet to red color, and more or less all these chlorophyll molecules or the pigment molecules, they absorb these lights in different wavelength, and of course we are looking for that the green plant, because the etiolated plant, how it becomes green. So in that case, is green plants nothing but they absorb all the white light and reflected back the green light, so that we are seeing the green things of the leaf. And if you are looking at the chloroplast, because we will be talking about these portions today. So in that case, is these have, as you know that the inner membrane have a stacks of your thylakoid stacks, and these thylakoid stacks, they have the membrane. So, and these membrane, they have the light harvesting chlorophyll complex, because this chlorophyll complex, as in the previous lecture you have seen that the magnesium, this part is common there, phytol ring is there itself, and all these rings, these hydrocarbon tails, they are very much flexible in nature. But in the same cases, if you are looking into this, so as a result, what happens, this long nonpolar hydrophilic tail, this is generally anchors into the lamellar membrane, and head portions has the complex ring with the magnesium at the center, which generally where the light emerges is trapped here. So this is the machinery of the light, your trapping system, and these portions is attached to the membrane, so that we can have this chlorophyll molecule, which is generally acting as a molecule which globally produce the foods for us. So this is the normal things where we are looking for that. And if you are looking the absorption spectra, that we have the chlorophyll A, this chlorophyll B, and then we have beta carotene. So this A and B, they have a region of this red region, whereas the blue or green region have the beta carotene, because this beta carotene perhaps act as a molecules where generally scavenge some of these A and B molecules perhaps are protected this one. So as a result, we can see that, because if you are going for a biochemical analysis, we will be having a large number of chlorophyll molecules where chlorophyll A, B, then C1, C2, C3, D, F, and everywhere these heads are there itself. Only there are differences in the tail region. And of course, if you are looking for the carotenoids, where the xanthophylls and beta carotenoids are there, and so they are the linear structure, whereas phycobilinoproteins, if you are looking into that, so where the phycoerythrin, phycocyanin, and phycobilins, they are the components, they are able to do the photosynthesis in our spectrum. So if you look at the history of this evolution of this chloroplast, how it comes, because cyanobacteria developed oxygenic photosynthesis about 2.6 to 2.5 billion years ago, very, very long back. And approximately 1.9 billion years ago, after the endosymbiosis, generally that cyanobacteria is placed in the host cell, so that in the evolutionary process, the plastid has emerges in the eukaryotic system. And about 100 to 200 million years ago, red algae and rhodophyta, as well as they have the common ancestry with these previous. And we could see the green algae, because they are perhaps this 100 to 200 million years ago, we could see that the same kind of pigments are there in your red algae as well as brown algae, whereas green algae appears in 1.1 billion years ago. So you start from that 2.6 billion years ago, then come to the 1.1 billion years ago. So what is the long space where the evolution takes place, and the chloroplast has emerges, and that gives us the food right now. But if we are looking back, because I several times told that our age in the earth and the earth ages and the evolution, if you are calculating in terms of Darwin's or mutation, it is not matching. Earth age is quite younger as compared to the human evolution. So as a result, from the single cell of Chlamydomonas to the very big tree, angiosperm, so it is quite astonishing to understand what exactly it is, and sometimes maybe we are implanted here from some other planet, it may be, we do not know. Okay, so if you are looking to the terrestrial plant, so where the carotenoids in the first positions, where generally they are absorbing the green lights as well as to some extent the blue lights, and then it can percolate it up to the chlorophyll B, then up to the chlorophyll A, where the chlorophyll A and B also can take this red light, also can take care by this chlorophyll A and B. So we have a reaction center, and chlorophyll A having these both PS1 and PS2, where this P700 and P680 is there. Then marine habitat, if you are looking to that, this brown algae and dinoflagellates, so generally it has also the carotenoids, then chlorophyll C, then chlorophyll A, and also have your PS1 and PS2. Then if you are going for a cyanobacteria, so in that cases fur reds are also absorbing by this, and some of the yellow lights also absorbing by the PC, then APC is by the red and fur red, then chlorophyll D is there itself to take care of, because all have the this your magnesium rings are there. And of course, chlorophyll Ds are there, and some other unknown products are there. So this, it has also the reaction center, and of course, cyanobacteria. When we are looking for that, so during that time, the chloroplast structure is not exactly this normal chloroplast structure of this angiospermic plant. So, and if you are looking into this another cyanobacteria group, so where the your bilinoproteins are there, then chlorophyll F, then chlorophyll A, and so it is as similar of this one, and then also it has a reaction center to manage both the PS1 and PS2 system. And radialgae, say for example, so where also we have because in this process generally we are missing this chlorophyll A part, sometimes chlorophyll B or C or F part, also plastocyanins are taken care of, and we have the photosynthesis. So as a result, what happens that all these cases when the chlorophyll A and B and C are there, most of the major chlorophylls are generally chlorophyll A and chlorophyll B, and formula wise they have the differences, and chlorophyll A possesses the methyl groups, whereas the chlorophyll B possesses aldehyde group, and it is essential pigment chlorophyll A for the photosynthesis, whereas the B and the other carotenoids etc., they are called your accessory pigments which absorb the energy of the different wavelengths and transfer it to the resonance or to the chlorophyll A, so that the chlorophyll A is supported by the chlorophyll B or the carotenoids or any other pigments which is there itself in the hierarchy. And of course, the chlorophyll A spectrum, if you are looking for that, of course, they have a maximum spectrum near about this 650 to 1700. The red region as well as the blue regions also will be having someone, and chlorophyll B and carotenoids, if you are looking into that, so that is generally in the blue and green as well as the violet portions it is covering. So as a result, what happens, both the chlorophyll A, chlorophyll B and carotenoids, they are having the major role to play there itself. And of course, everybody has this ring structures which have the much more similarity with this hemoglobin, where only the iron is replaced by this magnesium. And in the last lecture also we talked that how it is the similar structures is operated there. One is carrying the oxygen, whereas this is for your photosynthesis. So, and chemical compositions, if you look into that, proteins we have about 35 to 55%. So that's why I am very much I will be very much happy that after this lecture, if you are taking much more of plant, so you will be getting a lot of vegetables here, and here maybe you will be having this, you will be visiting this facility in the top, where they are also producing the proteins, this LSC1 complex and LSC2 complex as a powder, and they are selling in the market. So okay, so these lipids and you will be astonished that they are from even the larva of the any of the larva. Okay, although you are vegetarian, some are vegetarian, but it is larval protein. And sometimes the cabbage, the waste leaf of the cabbage, they are collecting and isolating this LS1 and LS2 complex. So that's why this is the protein which is marketable, and they are going for that one. And lipids, if you are looking into that, because we will be talking about the lipids 20 to 30%, carbohydrate about 3 to 7%. So that's why nowadays a lot of people have the diabetes in our country. So the carbohydrate, very less carbohydrate are there. Only the proteins you have much more. Chlorophyll about 9%, so dry weight, and chlorophyll A, out of that total chlorophyll of 9%, about 75% are chlorophyll A, and chlorophyll B is 25%. So chlorophyll A is much more there. And of course, to support that one, carotenoids we have 4.5% and nucleic acid 3 to 4% in the normal positions. So this slide is quite vital for today's lecture, the major glycolipids in the thylakoid membrane. If you are looking into that, yes, they are very big names, but later on we will be accustomed with this. So MGDG, then DGDG, SQDG and PG, these are the four things. They are much more vital glycolipids in the thylakoid membrane. And if you look into this, that MGDG, it is mono, it is di, and it is sulfino, a sulfur containing one, so that SQDG, so they are your glycolipids, whereas phospholipids is your PG is your phospholipids. And then if you are looking into these uncharged and negatively charged anionic proteins, so they are, these two are your SQDG as well as PG, they are your negatively charged or anionic proteins, whereas uncharged proteins are MGDG and DGDG. And non-bilayer forming is your MGDG, whereas bilayer forming, all the three are bilayer forming. So as a result, you can see that these are the four lipids, the glycolipids, those are the major component of the cell membrane, which is going for that. So your MGDG and your DGDG, because all these terms will be coming very frequently in the next slides. So, and if you are looking the membrane compositions of the plant plastid and cyanobacteria, thylakoids, MGDG, we have about 53%, so whereas DGDG, we have 23%. So monolipid, dilipid is very less there itself, inner envelope 49% and 30%, and SQDG 5% only, PG 8%, outer envelope about 17% of mono, di is 29%, and your SQDG is 6%, and non-green plastids, those are there itself, because sometimes that's just few minutes back, the lecture was that how this non-green plastid become green, and etolated plastid can be green also. And I just before the introduction, I was asking to Prasad that in the seed itself, the embryo which is there itself, it has a cotyledon, and that is not green color. Then there may be some plastids are there, non-green plastids are there, and during that time, whether all the enzymes, whatever you have talked about in the intermediate enzymes, they are there itself, they are activated or non-activated condition, or after the putting of this plant in a suitable environment, they are activated in the nuclear genome, and then this has a crosstalk into the chloroplast or leucoplast, and then become chloroplast. What is your answer? Because in dormant conditions, the plant having the cotyledon, which do not have any chlorophyll, so must have non-green plastid there, and during that time, all the greening process, whatever the enzymes are there, so all they are present there itself, and then it is finally activated, or there is a mechanism of transcriptional regulation which can trigger after the environment suits to germinate that plant, and then the etoplast or leucoplast become chloroplast, and during that time whether the chlorophyll is synthesized or not. So these are the questions, very basic questions, and you have to answer. We are old enough, we are not able to give answer. Only you are the researcher, you can give the answer to us. But anyhow, so that's why you can see that there are variations of, because major portion of this your MGDG is much more in the thylakoid, and then after this outer envelope very less amount, whereas this DGDG is more or less same type, and whether the MGDG is converted to DGDG or not, that is a question, and or it is converted to SQDG or not, or all these three are essential for not. So in that experiment, because professor was talking about that, they will be having some other talk, that meta-analysis, because if you are silencing some of the genes or knock out some of the genes, and then only you can have the effect that what type of your deficiency they have. So as a sorry, back how it could be this one. Yeah. So if you are, because to give a normal idea, theoretical idea about the PS1, your complex, because this is a computer generated complex of side view and top view, where the monogalactosidal diacyl glycerol or MGDG is present. So these colors, where it is there itself, they are MGDG. Then DGDG is digalactosidal diacyl glycerol. So they are your this color, and all these red color balls, small red color balls, they are oxygen molecules which is attached there. And then PG is also there itself in this blue color. This SQDG is in blue color, and green color we have PG. So these complex, all these glycoproteins, they are embedded into the structure itself, so that a PS1 systems can produce or in PS2 systems where the dimer formation takes places. So it's a monomer, and then dimer formation takes places. In that cases, we will be having a lipid molecules where 20 lipid molecules are there, and out of the 20 lipid molecules, 6 MDDGs and 5 DGDGs and 4 SQDGs and 5 PGs form this complex. So in that complex, what happens? The polar head groups of all the SQDG and PG molecules faces the cytoplasmic site of whereas those of the MGDG and DGDG molecules, except one molecule, they face into the luminar side. So that means topologically these attachment is quite interesting to see, because you have to visualize the things, so what it is going on there itself, not theoretically all these 3-4 terms. So in that cases, and of course, if you are looking the locations of these 3 PG molecules, so there generally we have the PG664, PG649, PG702 located near the QA binding site of the PS2. So in that cases, generally the oxygen, phosphorus and carbon atoms shown in the red, purple as well as green color respectively. So in that cases, what happens that we have different molecules, and D1 protein, D2 proteins are also there. It is in a specialized folding. So mostly it is of alpha folding. And then in the locations of 2PG, if you are looking into that, so there are also 3 protein molecules located near the QBB binding sites of the PS2. So as a whole, you can have a things that because we have talked about the chlorophyll, then inside the chlorophyll the different components are there, and in PG experiment generally we used to do anthocyanin or carotene and chlorophyll. But inside the chlorophyll also there are a lot of things to do, and also HPLC can give you the better solutions of each and individual component. And component wise, when you will be going for any of the experiment to do, so in that cases you need 2-3 things very important. Mostly those are doing the MSc, at least I am telling for them, that to learn the SDS page, to learn the isolation of the chloroplast, to learn the spectrophotometry, where this fluorescent your detector should be there, so that you can analyze the peaks, very small peaks also, and what exactly it is. And to get that one, of course, you have to need to take the help of the biochemist as well as chemistry people, that these small peaks of the compounds, what is the structure and how it is assembled with these. That's why in this course I think there are some computational biology which is also involved there, and which is very much lacking in the UGC syllabus, because as I know that all the UGC syllabus do not have much more of this one, at least for the botany people. Life science people, biotechnology, they have bit. Okay, but anyhow, so in these cases, so glycolipids in the thylakoid membrane of the spinach, if you are looking into that, so chloroplast thylakoids, this bar is your chloroplast thylakoid, then chloroplast inner envelope, and there are a lot of variations there where the thylakoids have the much more of about 55% of the chloroplast, whereas inner chloroplast they have a variations there itself. So the variations of the MGDG, DGDG, and SQDG there we could see, and variations generally in the mono we have lot of variations here. DGDG do not have much more variation, it's a constant one, whereas SQDG also not that much of significance. Of course, the PC has again a variations there. So plastids, that's why have a specific role in support the plant growth of life cycle that involves the synthesis of the lipids, amino acids, purines, and pyrimidine bases, and various hormones and secondary metabolites, as well as the nitrogen and sulfur. So you see this particular membrane structure, they have lot of functions to do in the chloroplast, and chloroplast membrane is unique lipid compositions with non-phosphorus mono and galactosidyl diacyl glycerol, so MDDG and DGDG respectively, and about 50% of MGDG and 25% of the DGDG of the total thylakoid lipid membranes we could see, and rest are comprised of anionic glycerol lipids and sulfo SQDG as well as the PG. Generally we have after this, out of this 50% of the MGDG or 25% of the DGG, rest are your SQDG and PG, and it's so that's why, and also with a marginal proportions of the your phospho hydrodidyl cytol, it is there itself. So, and also the glycolipids in the main constituents of the envelope of the chloroplast and non-photosynthetic plastids, with glycolipids about near about 60% of the total lipids we could have. And also these glycolipids and your SQDG are the minor constituents and are absent in the extra your plastidic membranes of the your plant cells, and chloroplast and cyanobacteria reflect the same origin. Because of that, the common ancestry of these particular components are there both in the higher plant as well as the lower groups. Of course, the pathway of biosynthesis is different, and these highly conserved lipid compositions among the chloroplast and cyanobacteria suggest a special requirement of these lipids for the oxygenic photosynthesis. And of course, there are glycolipids. That's why that in the thylakoid, glycolipids plays an essential role in providing a lipid bilayer matrix embedded with photosynthesis protein and cofactor complex. So that's why the assembly of these cofactors as well as the other components is quite important to have, not only the synthesis of these things, and coordinated biosynthesis of the thylakoid lipids with the chlorophylls and photosynthetic proteins during the chloroplast biogenesis. That's why it is very much essential to construct the thylakoid membranes network, which is generally so that we can have an efficient photosynthesis system. So that's why we could see that because this how these lipids generally help in the thylakoid biogenesis system. So in these cases, in the thylakoid glycolipids that provide the lipid bilayer matrix as well as the protein cofactors, generally forming these photosynthetic electron transfer systems, and modifications of the lipid compositions if you have, So that strongly affect the structures of the thylakoid membrane. So as a result, what happens, the thylakoid membrane, if it is lost, or if it is not in a proper stage, or in the developmental positions also, thylakoid membrane productions is a very vital essential things where the glycolipids plays a very big role. And crucial lack of the MGDG and DGDG in Arabidopsis cases generally causes no or severe underdeveloped internal membrane structure in the leaf plastid. So as a result, what happens that if you are devoid of, or if you have a mutant, so in that cases we could see that there are not much more changes, but the partial deficiency of the MGDG resulted in decreased amount of the thylakoid membrane. So as a result, stacking of the membrane can lose, and DGDG contributes a membrane stacking via the formation of hydrogen bond between the polar heads of the adjacent bilayers, and MGDG has cone-like shapes because it has a special shape which has a small head group and which allows it to form an inverted hexagonal phases, where the DGDG has a more cylindrical shape. So in that case, the protein shape and structure, 3D structure of the protein is also varying because of the component, and the ratio of this bilayer forming DGDG and non-bilayer forming MGDG affects the property of the chloroplast membrane by affecting the lipid bilayer for the hexagonal to the laminar phase. So as a result, hexagonal structure of this protein is collapsed because of this, if there is a variations in the DGDG and MGDG balances. So it is tightly regulated in the response of the various stresses, because when we are putting lot of stresses during that time, there is a changes in the DGDG and MGDG level. So as a result, that affects the size of as well as the protein structures of the chloroplast, so that the functionality of the chloroplast remain strict. As a result, you generally in normal cases we could see that decrease of chlorophyll content in normal cases we could see, but internally we have the changes in the MGDG and DGDG percentage. And also, if you look into this, that acidic lipids with negative charges in their head groups PG and SQDG also affect the organizations of the thylakoid membrane, and PG has a crucial role to in the proper thylakoid biogenesis, and deficiency of the chloroplast PG in the Arabidopsis, we could see that that formations of the vesicles, enlarged vacuolated structures generally we could see, so that this stacking of this your thylakoid membrane generally disturbed, and or we can see that underdeveloped, when the plant is growing during that time, underdeveloped membranes generally we could see because of this deficiency of the PG in the chloroplast level. So that's why in that previous slides we have seen that there are variations of the content of the PG level there itself, and in ratio of this bilayer forming DG in non-bilayer forming MGDG affects the properties of the chloroplast membrane by altering the lipid bilayer from the hexagonal to your lamellar phases, that also we have talked just in the previous slide. And also it is tightly regulated in response of various stresses, that is also there, and also your deficiency of the SQDG causes the strong curling of the thylakoid membrane. So as a result, the lamellar structure of this thylakoid generally affects and generally grana stacking have little effect, but in the Chlamydomonas, but in the higher plants we have that type of problems in the SQDG deficiency. And lack of SQDG biosynthesis in the Arabidopsis had no effect in the thylakoid architecture. Sometimes we could see that type of phenomena also. Some of the papers they have shown. And loss of SQDG together with a small reduction in the PG content decrease the grana stacking in the leaf chloroplast. So that means that SQDG and PG, they have a correlation between, so that that generally affect individually, may not affect, but combinedly they have the effect in the grana stacking. And thylakoid lipids, that's why, are required for the structures of and functions of the photosynthetic complex, beside providing a bilayer matrix. And X-ray crystallography of cyanobacterial PS1 and PS2 revealed the both complex involved in the thylakoid lipids in their crystal structures with MgDG and PG near the reaction center of both the cases. And PG deficient mutant in the cyanobacterium, we could see that Chinocystis PCC 6803 demonstrated that PG is essential for the PS2 electron transport from primary plastoquinone QA to secondary QB. So in that cases, electron transfer is severely hampered because of this PG deficiency. So that's why PG also a equally important part of this lipid biosynthesis process. And also the stabilization of oxygen evolving complex by the extrinsic proteins and terminations of this PS1 complex, also it has a effect of this PG, and PG is required for the chloroplast development and photosynthesis, and which is generally found in this Arabidopsis as well as Chlamydomonas mutant. And another anionic thylakoid lipid, that SQDG, is crucial for the activity of the PS2 at both donor and acceptor side in the Chlamydomonas and your Chinocystis, which is generally experimented there itself, and it does not have much more effect in the PS1 activity. So SQDG also is not required for the photosynthesis of biogenesis in the Arabidopsis under the nutrient sufficient condition, where the Pi is much more, and this is required for the maintaining of the amount of the total amino acids in the chloroplast when the PG content is decreased under the phosphate deficient conditions. Generally, we could see that kind of situations. So requirement of SQDG for the photosynthesis differs among the organisms because of that P deficient condition, phosphate deficient conditions, and in the normal conditions we could have different type of effects. And of course, DGDG deficient mutant, generally DGDA. So in that cases, this is the gene which is responsible for that one of the Chinocystis species, shows that DGDG is required for the PS2 activity by binding the extrinsic protein that stabilize the oxygen evolving complex. So that the complex, the binding of this oxygen evolving complex as well as the protein, where the DGDG plays a very big role there itself. And of course, mutations of that DGDA gene, which is generally sensitive for the photoinhibitions under the high temperature, high light, and repair cycles of the photoinhibitions more severely affected in that cases. So what happens that if you are having a stress of high light or high temperature, and during that time, so this your DGDG generally got affected, and so that we can have a differences. And we could see in this forum, perhaps it is very much not directly related, but indirectly related for our global population right now, because in this state or major state of the India, mostly southern part, they are taking the rice, and we are expecting about 663 ppm of carbon dioxide in 2050. So after 25 years, we will be expecting that one. And temperature rising will be about 3 degrees more in the natural condition. Whatever the temperature it is there, it will be rising. So in that cases, we were just given a task by the central government that what type of paddy can be grown in that high carbon dioxide concentration as well as high your temperature. We just screen about 63 varieties of rice, and out of them we could see that there are, because of course we have not done this chlorophyll part, only we are concentrating in the rubisco part. And we could see that only 2 varieties of the rice, they have sustained this rubisco, and they have very good activity, at least up to the 3-degree temperature, higher temperature, as well as your so you can imagine that in our country, if you have that type of scenario, that only 2 varieties will be performing after 25 years. During that time, I will not be there, you will be there, and your child will be there. Okay, you will see that. So only 2 varieties will be performing, and the rest variety is not performing. What they are doing? Because the photosynthesis, when it is, so it is actually converting your photosynthetes or the glucose to the starch, so that we could see that in higher carbon dioxide concentration and high temperature, the biomass is very much increased, but the grain filling, it is not happening. So that's why we have chappy rice. So that's rubisco is not working properly. So that's a less conversion of that one, and of course there are some other genes which is responsible for that. And so in that cases, it is the best forum. You try to practice rice leaf to eat, because there is the protein and everything is available. Instead of rice, starch, you just go for your chewing this rice leaf, so that you will be getting the food as well as anything, just like the cow. So in that cases, you have to take the help of this your biotechnology people to isolate the protein, and then take the protein in a liquid or in a glass of water, like that. But anyhow, so that's why they have, because sometimes it is a because the till date, whatever the variety we have released by this ICR in the breeding program, so not a single variety is trustworthy for giving this one. And we could see that there are 2 your LSC genes are responsible for there, and which is generally available there itself for transporting the silicon from the soil. The silicon they are transporting and keeping into the rice leaf, and this transporter gene, they have changed there itself, just like a maize. In this particular 2 variety, those are surviving with your your rubisco. We do not know yet what is the connections between the rubisco and your transporter gene of this your what you call it, silicon. Anyhow, so, and also if you are looking for that, DGDG also plays a role in decreasing photoinhibitions under the high light, because if there is a high light conditions during that time, of course this photoinhibition takes places, and DGDG also require for the terminations of the LHC2 complex and accumulations of the galactosidal diacyl glycerol in the DGDG deficient mutant could not fully restore the PS2 functionality, because the dimerations of these LSC2 complex generally hampers. So as a result, galactose moiety of this DGDG may be important for the increase with the photosynthetic protein complex of DGDG. So as a result, what happens, we could have that conditions, and 40% of the decrease in this MGDG in Arabidopsis mutant has no effect in the intrinsic PS2 activity, but impaired thylakoid membranes are generally found, and loss of MGDG decrease the level of the cytochrome B6F complex. So as a result, MGDG also have a big role to play into this cytochrome B6F complex, and it is blocked the intersystem electron transfer in the tobacco, so which is generally also found here itself, because in the OGIP analysis also we could see that there are blockages sometimes in different protein complex, and about 80% of the loss of the MGDG in the partial reductions of the DGDG strongly impair the pH2 photochemical reactions. So it disrupts the coupling between the reaction centers and antenna complex. So as a result, what happens that it has a major role to play. MGDG also has a major role to play in the coupling reactions of these reaction center and antenna complex molecules, so that the effective electron transfer is not proper because of the loss of MGDG in the membrane. And it is also synthesized this because MGDG is synthesized by the MGDG synthetase enzyme, which is transfers the galactose from the UDP galactose to the diacide to glycerol in the plastid envelope. And out of this MGDG synthetase, there are some homologous genes which are responsible for that, and they have also the different effect in that, because generally the MGDG1 is the have the major role in the galactolipid synthesis, whereas 2 and 3 are localized in the outer envelope membrane, constitute of alternative pathway for the P limit condition. So when the P limit conditions are there, or deficiency of phosphorus, so during that time they will be taken care of instead of MGDG1 gene. And being a binding block of this chloroplast membrane, generally MGDG is used for the DGDG biosynthesis as a substrate there. And so this is the whole scheme of the productions of PG, then TGPG, then your because this is another path where also the SQDG also can be produced. So as a result, this total biosynthetic pathway, if you are looking into that, there are several genes which are responsible for each and individual steps, and where mostly the block the what you call your things bolts letters, they are nothing but the enzymes which is responsible for that one. And in that cases, DGDG biosynthesis is primarily catalyzed by the DGDG synthetase, which transfer the second galactose to the MGDG for the EDP galactose in the plastid outer membrane. So this process generally this DG1 and DG2 found in the Arabidopsis or in other angiosperms, and DGD1 catalyzes the bulk DGDG synthesis in the chloroplast inhibitions with the MGDG1, and DGDG2 functions in alternative pathway of the MGDG2 and MGDG3. So as a result, what happens that the DGDG biosynthesis generally also get affected if there are some changes in these components there itself. And another enzyme also sensitive for the freezing 2, that SFR2, that is endemically different from the DGDG synthetase, which functions from that DGDG and oligogalactolipids. So it is located into this SFR2 enzyme is located in the plastid envelope membranes and transfer the galactose head group from MGDG to a second MGDG or to a oligogalactolipid that increase the number of galactose moiety into the polar head. So, and it does not contribute significantly in the DGDG synthesis during the normal growth, but is required for the freezing tolerance to absorbed by this plant. So during that time, also this SRF2 are responsible for managing whole the process of chlorophyll assembling. And also this in this process, the PG synthesis, when takes place in the plastid and mitochondria and endoplasmic reticulum in the plant, and the first step of the PG biosynthesis conversion is phosphoric acid into the CDP diglycerol by the CDP diglycerol synthetase enzyme, and 5 genes are responsible for that. In that case, CD, CDS1 and CDS5 encode the functional CDs in the Arabidopsis genome, whereas 1, 2, and 3 are targeted for the to the endoplasmic membrane, endoplasmic reticulum, and CDS4 and 5 are located in the plastid and functions for this thylakoid membrane biogenesis. So as a result, what happens that we could see that also there are functions of this PG synthesis in the membrane also, and the last one is your SQDG synthesis in the plant. If you are looking to that UDP glucose is synthesized from the glucose 1-phosphate and UTP by the UTP glucose phosphorylase 3 in the plastid. So UDP, your SQD synthetase converts the UDP glucose to the UDP SQ with the sulfide. So as a result, finally you could see that this SQDG generally increases in the response of the P deficiency system in parallel with the decreased PG content, which may help in the proper balance of anionic charges thylakoid membranes protein of this one. So as a result, if you look into this, some of the results we have taken from some of the papers, that these chlorophyll accumulations in the PGP1, 2 with the P deficiency, P1 deficiency, that we could see that there are changes in the chlorophyll A, chlorophyll B, and chlorophyll A by B ratio. So of this wild as well as the mutant, and also morphology and diameter of the plastid nucleoids of the protoplast from the wild type as well as PGP1, 2 levels under the PI and non-PI conditions. Generally, we could see that there are nucleoid formations, which is these small fluorescent particles correspond to the plastid nucleoids in addition to the large fluorescence from the nuclei. And we could see that there are variations in the nuclear diameter also. So as a result, what happens that conditions of your different glycolipids also can leads to the formations of different size of your nucleoids into the chloroplast, we could see, and that has been supported with these because in the wild type, if you are looking to that, because this is Kobayashi et al. 1914. So Plants report, they have reported that this wild type, they have D1 enhance D1, then D2 also enhance, LHCP complex also enhance there itself. But in the mutant, we could see that there are P plus and P minus mutant. We have the variations there itself. So less amount or even nil amount of this D1 and D2 protein as well as LHC complex. So that's why we could see that because all these theoretical aspects what we have talked about, that can be also experimented, and also these are all culmination of different papers. I really do not work that much in that particular subject. And also, if you are looking to the thylakoid lipid synthesis in regulation of the chloroplast biogenesis, that generally the photoreactive chlorophyll allows the plant to convert the light energy into the sun. It is normally for the chemical energy, and most of the cases MDGG plays a vital role in the xanthophyll cycle via the solubilization of bioxanthin and mediating the binding of this bioxanthin to the epoxy side to the thylakoid membrane, so that the hexagonal lipid structures formed by the MGDG in the thylakoid membrane is essential for de-epoxidation and of the xanthophylls, and therefore involves the photoprotective and non-photoprotective photochemicals quenching of the photosynthesis. So and also we could see that MGDG biosynthesis is essential for the thylakoid membrane biogenesis, And internal membranes of this biogenesis under the P deficiency condition includes the chlorophyll accumulations and partial formations of PS2 complex in the MGD1 to genes. Absence of this and photosynthetic electron transfer was still desulfonylated in the case of the mutant. So thus the lipid biosynthesis is essential for the nucleoid modifications during the thylakoid membrane formation, as well as biogenesis and expressions of photosynthesis associated genes in the nucleus, as well as the chloroplast and or the plastid. And also that regulated structures of these DNA and RNA polymerase complex, because that GUN1 gene is generally found which is act via the nucleus and generally for the thylakoid biogenesis that affects the expressions of the photosynthesis associated nuclear genes at the onset of the chloroplast biogenesis through the plastid signaling pathway, which could be independent of the photosynthetic activity. Because of that, MGDG12 and PGP12 shows no photosynthetic electron transfer activity under the P deficient condition. So as a result, what happens that gene which is acted and maybe some transcription factors which is regulated the genomic constituent and also the intertalk between this nuclear genome as well as the plastid genome to regulate this lipid synthesis pathway. So as a summary, if you are looking to that, more or less it is 110. Okay, so the concerted synthesis of the assembly of the membrane lipid and protein pigments and cofactors are required for the efficient photosynthetic system and chloroplast biogenesis, of course. And thylakoid lipid biosynthesis can trigger the expressions of the photosynthesis associated genes in both the nucleus and plastid. And fatty acid synthesis may be linked to the plastid protein synthesis. And so that the biogenesis of the thylakoid lipids is coordinated with the biosynthesis of the photosynthetic protein cofactor complex at the transcription level and post-transcriptional level in response to the endogenous and environmental causes. And of course, the reciprocal coordinations of this synthesis also there. Because of that, when some of the stresses become activated during that time, they are coping with the system with the help of some of the bypass route, which is also we could see. So that's why in a normal case, this is perhaps the last slide. So where if you are maintaining this chlorophyll A and B ratio, because if you are going for a engineering of any of the plant where the AB ratio, if you change, so in that cases you can have a plant where much more of your light harvesting system as compared to the normal one. So in the normal one, if you are interested for having much more of productivity, of course, grain filling, say for example, is a concern. So where we need to have entrap large number of your energy, and chlorophyll is one of the major component where the glycolipids are playing a vital role for stability of the membrane, as well as the LS1 and LS2 complex, as well as other coenzymes which need to be activated and of course well thought, so that we can have if we are engineering some of the plants having the chlorophyll A by B ratio, where it can give much more productivity. And thank you very much for your patience hearing, and perhaps you are late for 10 minutes for lunch. Any questions if you have, or I am leaving this slide to you, it is up to you. And anything, if any questions, I will be glad to answer anything if it is. জ্যোতি সেটা পানি আছে?
Sir, I have one question, sir.
Sure.
From dormancy to germination.
Yeah.
Thing that you told is very interesting. From etioplast to chloroplast.
Yeah, chloroplast. Yeah.
What I think is all the genes, they are already there
In the system. Yeah.
under favorable conditions, the transcription machinery or protein degradation process, activate those genes, chlorophyll biosynthesis.
Yeah. Yeah.
That is how chlorophyll is synthesized.
Yeah.
During after the germination.
Yeah.
Am I right, or is there any?
No, that may be the cause, because sometimes also you could see that if the chlorophyll also there itself, sometimes it become also leucoplast. Chloroplast become also leucoplast. Say for example, in a plant, the upper part is chloroplast, lower part is your leucoplast. Root have the leucoplast. So there is a transition where the stem and root transitions are there. There may be some glue anybody can work because some machinery is there, how it is switching on this leucoplast to chloroplast. Or say for example potato, if you are keeping outside, sometimes it is greening on the upper part. So how the leucoplast become greening? So this is some of the things which need to be, because I always told my student that you just go out from your this black and white writing, just go out from there and you think the nature, what it is going on, and take some of the questions, pick up, then digest. If it is not, then go to your sir, because sir is nothing but the gap between the two words of a sentence. One sentence, many words. If there is no any gap, you cannot understand anything. The gap is your sir, and gap is also answer as well as question. So if you are thinking much more, you will be getting much more. And always you try to think three-dimensionally. What exactly it is going on? So bilayer membrane we are talking about. In photograph, it is only a single layer. But 3D, what it is going on? Now the computational science, they are giving much more of your focus, and you can get much more of understanding process. And that's why sir was talking about that it is our duty to ignite. Out of these 30 students, if one is ignite, this is more than enough for us for this lecture. Thank you very much. Once more, thank you very much. And I profusely thank to Professor Rajgopal sir, as well as all the participants, as well as our Jyoti and sir also, that to give this opportunity. And yesterday, actually, I could not make it because of that. We have a crucial problem in the university that in the +3 level, we required a question day after tomorrow. And 10,000 students are halted. So the message has come from the Vice Chancellor that you just prepare the things immediately you send. So yesterday night, I just prepared that one, sent that one, and then I came here. And tomorrow maybe I have to leave to Bhubaneswar. So thank you very much, and wish you all the best for this fruitful your training, as well as future of your everybody's life. Thank you very much, and once again thank you, sir, for nice meeting you. And maybe sometimes if we have actually, if we could know earlier, so I could connect you to take to our university. And the next time when you will be coming, it is our wholehearted invitation that you should visit us. Okay? Thank you very much. Thank you very much. Thank you very much. Thank you very much.
Thank you, sir, for the